be available. If you have any issues with being on a recorded webinar, you are welcome to leave. Your face will not be shown. Thank you again for joining us today. We will be welcoming our speaker in just a few minutes. I just want to start and talk a little bit about the Dan River Basin Association. We are sponsoring this lunch with DARBA today. DARBA is our acronym. We serve, our service area is about 3,300 square miles, including 16 counties in Virginia and North Carolina. It is everywhere the Dan River flows and all of its tributaries. The Dan River starts up here in Patrick County and flows all the way down here to the South Boston, Halifax area and crosses the Virginia, North Carolina state line 8 times. We protect our rivers and streams and our natural and cultural resources through 3 major goals: recreation, education, and stewardship. We truly believe in the philosophy that when people get out and they experience the mind, body, spirit benefits of being in nature, they want to learn more. And so we have a lot of educational programs about the outdoors, and our natural resources, our cultural resources. And then once they learn more about it, they want to protect it. And that is where our stewardship goal comes in. You can learn about all of our programs and our services that we provide on the Dan River Basin Association website. It is danriver.org right here at the bottom. Please help us preserve and promote the wonderful and unique resources of our region. And with that, I am going to introduce Dr. Jackson Means. Jackson, you want to turn your video on? There he is.
Hello.
Dr. Means is responsible for curating and cataloging specimens from the Virginia Museum of Natural History's Myriapod. Did I say that right?
Good.
Holdings. It is one of North America's premier collections. Among other responsibilities, Dr. Means conducts primary research on millipede taxonomy, systematics, and natural history, as well as collects, preserves, identifies, and curates those specimens. Dr. Jackson's love of nature began early. He grew up in Keswick, Virginia, tromping around the woods and playing with bugs. And then when he was in college, he informed me that that love of nature turned into an interest in species discovery. He graduated from Denison University and went on to graduate school at Virginia Tech. I think he found his true passion there and began working on millipede species discovery in the Appalachians for his doctorate. He's published a lot of good stuff out there about collection techniques, taxonomy, species description, and he is always very excited about collecting millipedes on private land, he tells me. And so if you own any property and you would like to know about the millipede species that are there, you can reach out to him at the Virginia Museum of Natural History. And he might come and visit you and discover maybe a new species on your land. So with that, I am going to turn this over to you, Dr. Means. And thank you so much for giving your time today and educating us about these very interesting little creatures.
Well, Tiffany, thank you very much. I'm excited to be here. Let me go ahead and switch over. So today I'm going to be talking about myriapods, which are millipedes and centipedes and their relatives. And I appreciate you all taking the time out of your lunch to watch. Unless you get creeped out by creepy crawly critters, there shouldn't be anything in here that's too disturbing. I took out some of the grosser material. The insect world can be pretty brutal. So this should be lunch appropriate. All right, so for an outline of the talk, I'm going to start by talking about what the Myriapoda are, some biology and ecology, and then some Appalachian and Piedmont millipedes that you might find if you're going out hiking or gardening or anything. Then I'll talk about the collection here at the museum and my research. So the two less well-known groups in the Myriapoda are Parapods and Symphyla. Parapods are little, basically Twinkies. They're tiny little Twinkie-looking creatures they can be half a millimeter. So really, really tiny, soil dwelling. You find them under leaves if you look really hard. They have two branched antennae, which you can see here. It's a very unique characteristic. They have this weirdly branched antennae. There's about 800 species. So, people do work on these, but they're fairly diverse. Class Symphyla is a little bit less diverse, about 200 species, but they're larger. You'll definitely see these if you garden, and they're called garden centipedes by a lot of people, but they're not actually centipedes, but they are related. They're really fast. They're white. Their antennae kind of go like this real quick, and they'll run around in the leaf litter, completely harmless. But you'll see those a lot. They're very, very common. And then the two big classes that everybody knows in the Myriapoda are the Chilopods and the Diplopods. Chilopoda are the centipedes. They can be up to 300 millimeters long, so those really big Scolopendramorph centipedes you see out west or in Asia or South America. But they can also be pretty tiny, about 5 millimeters. They have one pair of legs per body segment, as opposed to millipedes, which have two pairs of legs per segment. That's the big diagnostic character that's easy to tell these two apart. But another thing is that centipedes are predators. They're very fast. They have venom delivering. They're not actually jaws. They're modified front pair of legs, but you can kind of see it here. But they deliver a powerful bite and a toxin. Millipedes, however, are detritivores. They just eat leaf litter. They're kind of like cows of the forest. Pretty harmless, but they do often have chemical defenses, which are not a big issue for humans. But I do want to point out that diplopods are much more diverse. There's about 12,000 species in 12 orders, as opposed to 3,000 species of centipedes in 5 orders. So we'll go into centipedes real quick here, because my expertise is on millipedes, so I'm going to focus on those. But for centipedes, you have 4 extant orders. The Scolopendramorpha, which are the ones I mentioned that have the gigantic, they can get really big, and they have these really big jaws, and they can give you painful bites, but they can be colorful. They're, I'm not trying to paint them as some evil thing, but they are aggressive. They can be aggressive. So try not to handle Scolopendramorphs. Luckily around here, we don't get anything bigger than this fellow here. I think this is a Scolopocriptops, which is it can give you a bite, kind of like a bee sting. Lithobiomorphs are rock centipedes. They also can give a nice bite, but they're not, they don't really get too big around here. Geophilomorphs are really thin, small, leaf litter dwelling. They're not going to bite you or anything. You'll find these if you garden, and they're really diverse and very beautiful, actually. This is a picture I took of some geophilomorphs from Puerto Rico, and they're just all sorts of colors and really pretty. And then there's the Scutitiomorpha, which everybody gets freaked out by house centipedes. This is actually, they are really cool organisms, fairly harmless. They're not aggressive at all towards us, but they do keep cockroaches and things, pests down in your house. So they can be beneficial to have. However, a lot of people find them in their bathtubs and stuff, and that's not super great. They're kind of creepy. They are very, very fast. Their hind legs actually move in front and over their forelegs as they run. So they have this really weird kind of gallop that makes them go very, very fast. They also have prehensile tarsi. So instead of having just simple claws like these guys have at the tips of their legs, they actually have these filiform beads that they can wrap around like a tentacle around things. They also have complex compound eyes, as opposed to very simple eyes of the other centipedes. They have much more complex eyes. So they're really, really fascinating. They are not native to the US, though they've been here now for so long that a lot of people, there are naturalized populations that you can find living in the wild, but they're not originally from here. All right, so millipedes, class Diplopoda. Most of the segments of the body, this is kind of the characteristics of millipedes. Most of those segments are fused into diplo segments, which is why they have two pairs of legs. So basically 2 body parts fused into one. And they can have as few as 12 legs up to 750 pairs of legs. The name millipede would imply 1,000 legs, but that's kind of just a misnomer at this point. There has not been any published millipedes with over 1,000 legs. The true millipede. Their mouth parts are typically modified into a grinding plate, which they use to break down organic matter in the forest floor. Their antennae are clubbed with 4 sensory cones. They are detritivores, and they are usually chemically defended. They produce chemicals. And this is Apalachiaria separanda calcarea that I got at my house in Shawsville, so fairly near this area. All right, so don't worry too much about all of this. I just basically want to point out the body of a millipede is broken down into a head and then a long trunk is what it's called. And then they have a telson, epiproct, paraproct, but basically the end of the millipede. So it's basically a long trunk, long body made up of a bunch of segments. And you can see the first 3 or 4, first 4 segments there just have 1 pair of legs. And then they start getting more, or the first 5. All right, so for us, the big species identifier that we use are called gonopods. These are the male reproductive structures. And so most millipedes, when you get down to a genus, like a specific genus, they all kind of look the same. And so you need to be able to differentiate them using something. And luckily, their gonopods are often really diverse. They can have all sorts of weird structures that are very interesting. And so gonopods are modified legs of the male millipede used for sperm transfer. And so the gono is sex, pods is feet. And millipedes are actually so ancient that they are the first organisms to have external reproductive organs. We have fossils with complete body plans of millipedes, which means they had to have originated earlier. But those fossils date back to around 410 million years ago. So they are believed to be, the first organisms to have breathed air on land, the first organisms to have chemical defenses, and the first to have external reproductive parts. And so this is what we use, kind of the shape of these gonopods, what we use to differentiate species. And this is just an example of a very different millipede from an Amphiloria. And it has, again, these very complex, interesting looking gonopods. And so highly variable. Females have what are called cytopods, which are a sclerotized it's like an opening down, below their neck, if you want to call it that. And it has it's basically a membranous sac that is protected by 2 valves that can vary in size and shape that open up to allow sperm transfer. It's very underrepresented in the literature, mainly because you have to dissect these to look at them. And so it's a lot more work than just the gonopods that are out there and highly ornamented. But we think that these contain taxonomic value. And so people are more and more trying to include these in taxonomic revisions, because there probably is a lot of information there that we're losing by not looking at them. All right, so for ecology of where millipedes can be found, they really prefer moist forests with plentiful leaf litter. Millipedes, for the most part, don't have a waxy epicuticle. So they don't have wax on the outside of their body like insects have. So they can't really regulate their water content as easily. And so they need to be in really moist habitats for the most part. There are millipedes that have adapted to deserts and grasslands and like really high up mountaintops. And some have actually adapted to urban areas. But the vast majority, and certainly what you're going to find around here, are going to be millipedes in kind of either swampy or gullies and rhododendron groves and really, really cool, wet places in the forest. I say typically detritivores here. They are all detritivores, but a few have been found on dead bodies of pigs and things like that, or found under those. And so it's still kind of up it's kind of debated whether they actually are feeding on animal matter or whether they just happen to be under the pig because it's a moist, habitat. But I wouldn't be surprised if they do consume animal matter, but the vast majority, I mean, they are just eating leaves and leaf litter, logs, branches, things like that. Mushrooms, moss, yeah. Find them eating lichen. They are everywhere in the world except Antarctica and very, very diverse in the tropics. However, Appalachia is a huge biodiversity hotspot for millipedes, and especially the family Zystodesmidae, which are the flat-backed, colorful mimetic millipedes that I study, or one of the groups I study. They are most diverse in the Appalachians. That's where we have the most species. All right, a lot of things eat predators. A lot of predators eat millipedes, as you might imagine, since there are so many of them and they're, slow moving on the ground, flightless organisms. However, millipedes, and I'll get into all the defensive mechanisms they've developed to combat these predators, they have a lot of defensive mechanisms. So these predators have had to evolve cool ways of stopping them or of catching them. So there is a mammal in Madagascar that has learned to smash pill millipedes. There are these millipedes that are the size of like a tennis ball sometimes, or smaller, golf ball to tennis ball, that are green. They're really pretty. They're actually arboreal in Madagascar, and they have a locking mechanism, so they can curl up into a perfect sphere and then lock, and they can't be pulled apart unless you smash them against a rock, which is what these mammals have learned to do. They smash them against a rock and then eat them. This is an ant, an antler jawed ant is what I've heard it called, but they feed specifically on the pincushion millipedes, which I'll explain. I'll show those later. But they're a type of millipede that does not have a hardened exoskeleton, and instead has urticating hairs all over them, like a tarantula. So they have these hairs that break off like a porcupine into an ant's face and gum up the jaws and cause issues. Well, these guys have evolved to they use these jaws like combs to basically peel off all of those hairs, so they can therefore get at the exposed millipede. And I did talk to one ant expert in Costa Rica when I was down there at their museum, and he said that he's actually observed those ants take those quills back to their nest and lay out like a palisade, sticking them in the ground as a protective mechanism for the nest. I haven't seen that published, but it was a cool story and I want it to be true, but I'm just giving you a caveat. I'm not 100% sure, but he said it was real and he's seen it, so that's pretty cool. Then these are Phingodids. I had a video of this and I took it out just because it's kind of gross, but these are actually adult beetles that are larviform, so they don't have wings. The adult females are larviforms. They look like big larvae. They have venom delivering jaws, and they will bite the millipede under the neck, paralyzing it so it can't release chemical defenses. And then they'll chew the neck off and go down the trunk eating, which gets pretty gross. Then there are salamanders, which definitely eat immature millipedes. So when they're really young, the millipedes don't produce chemicals quite as much. And so we've definitely found salamander gut content with lots of millipedes. And then there are hemipterans, true bugs that also feed on them. And there are phorid flies. I don't have pictures of them here, but there are flies that parasitize, lay their eggs in millipedes. So all sorts of predators. One group you may have noticed is kind of lacking were reptiles. We don't know really, there's not much evidence that reptiles really bother with millipedes, but this is a fence lizard I found at my house, and it was just so handsome I couldn't include it. I could not include it. And maybe it feeds on the millipedes that I have crawling all over the place, who knows? But he's handsome. All right, so we'll get into defense for millipedes. The number one defense that you notice right away when you uncover leaves and find a millipede is what's called vulvation, which is basically rolling up into a ball for defense, to protect yourself. And some millipedes are really good at this, like this spirobolid millipede. Others, not so great. Nanaria does a pretty good job of rolling up. They're very cute. Striaria here, they try, but they're very small. I don't know how much rolling up really will do for them. But almost all millipedes do some kind of like curling up. There are a few like Pseudopolydesmus and some other groups that run really fast, but for the most part, they're going to roll up if you uncover them. And then, as I said, most millipedes have defensive chemicals. And so this is kind of a simplification of what that would look like as a cutaway of the body ring here with the legs. They have what are called ozophores on the sides of their body, running down the body, that have these glands basically attached to them where they can secrete a chemical defense. And there are different types of glands, and they vary in complexity. Here's a picture of an ozophore under scanning electron microscopy. So they're pretty cool looking little openings. And so some creatures, like some millipedes, like Glandulus glutatus here, which is a invasive species, you can actually see the ozophores running down the body. It has kind of a transparent body, and then these red pigmented ozophores, which may be a warning signal to predators. So my lab mate at Virginia Tech, Derek Hinnon, worked on cyanide glands, and he did these great dissections that I'm going to show you here. And cyanide glands in the Zystodesmidae are the most complex type of gland because they have to mix chemicals in a reaction chamber before emitting hydrogen cyanide. They don't just carry hydrogen cyanide around with them. That would be dangerous. So they have the precursors. So they have a chemical reservoir here where they have precursors, and then they have a valve that opens up, and there's an enzyme that reacts or that is mixed into this reaction chamber. And you'll actually see that this reaction chamber here is a little thicker, so it can kind of take the sometimes kind of violent reactions of the chemicals. And then there's this is where the ozophore would go to. This is all really membranous. It's quite an impressive dissection, I have to say. He did a good job, but yeah, so that's the most complex, not super complex, but it's the most complex glands that you get in millipedes. Here's a picture of what that looks like. This is Rudaloria Clamputeri, which is a Zystodesmid you can find around this area pretty readily. And they produce, so cyanide is odorless, and it's like a gas, flavorless, odorless gas. So you'll smell like a cherry, really nice like cherry cola smell. Those are the benzaldehydes that come with the cyanide that tell you like, hey, I'm excreting something. But it also comes with this white kind of milky, sticky stuff that gets all over your face if you're like a mouse or something. And so when you pick these up, you'll smell kind of a pungent stink of well, it's really good smelling. It smells like cherries, but it is there's some cyanide in there, so don't sniff it too deeply. But yeah, you'll also see this kind of milky excretion on the what are called paranoda. These are the paranoda here, the edges of the body. Then there are millipedes that are what's called cryptic, and so instead of being flashy, which I'll get into the flashy, colorful ones later, a lot of them are really, really drab, so they blend into their background. This is a Nanaria from Ohio, Nanaria Terracola, and you can see it's only about, probably stretched out all the way. It's like, 20 millimeters or so, 25, and it blends right into the background. I mean, it's really hard to see. In fact, when I'm collecting these and going through, they often look like mycelia. If you just glance, it looks kind of like fungal mycelia just in the dirt. You don't really think anything of it. So they blend in really well, and that's a pretty good defense mechanism. Then sociality might be a defense mechanism. We think it is. It's not. Sociality or social behavior is not really common in millipedes, but there are a few groups that do it, including this group. This is Brachycybe, which are these feather millipedes. They're called. They're really, really flat, and they will get in groups and feed on fungus. They'll actually feed on these fungus. They'll get their heads all together. You'll find them under tulip poplar logs almost exclusively. And they all produce chemical defenses. And so we think by getting together a whole bunch of them, they're increasing that chemical defense if they're disturbed. So it is some level of defense to be social. All right, Apostemitism is bright warning coloration, and it's what everybody really loves about a lot of millipedes. A lot of millipedes are really, really brightly colored to show off that they are toxic to predators. So predators will learn to avoid that color pattern. And so these are two millipedes that are not from the US. This is from Malaysia, and this guy's from Africa. They're really, really beautiful though, so I wanted to show them just to give you an idea that there's a lot of variation. But here in North America, we have a lot of contrasting reds, yellows, and oranges with black backgrounds. That's kind of your common aposematic coloration. However, there are some blue millipedes in Knoxville, Tennessee. If you go to the forest behind the University of Tennessee, you'll actually find these gigantic blue Zystodesmids that really smell. I mean, that'll knock your socks off with the cyanide. But anyway, most of them are going to be yellow, orange, and black and red here. And so these are some common millipedes that you'll see. A lot of Aphiloria. We've got an Appalachioria here. And then we have Charocia is a really common genus, and it has a whole bunch of cool color patterns. So these are all out during the day. So you have a little sun here. These are all diurnal for predators that are out during the day and that can see in sunlight and are exposing, leaf litter to see millipedes during the day. There are also millipedes that have adapted to be aposematic at night. And so this group, Motixia, is out in California in the Sierras, and there's actually one in Los Angeles, in Hollywood, that area that is still alive, that's still surviving. But they are bioluminescent. And so like a firefly, they produce their own glow at night. And that is because they've evolved against, grasshopper mice. Grasshopper mice are these little hopping mice in California that are voracious predators. And so these guys actually glow, and I've gone out and collected them, at 3 in the morning or so. And it's really cool. They're not bright like a lightning bug, but they do glow. Once your eyes adjust to the dark, you'll see them just crawling along on top of the pine leaves or needles. It's really, really cool. Then there's also fluorescence. So fluorescence is different than bioluminescence. Bioluminescence is where you are glowing of your own accord. Fluorescence is where you're reflecting UV light, and a lot of millipedes fluoresce. A lot of them here, especially fluoresce. And so if you shine a black light, the millipede will glow, and it's really, really impressive. Kind of like a scorpion, if anyone's ever seen a scorpion fluoresce. We think that is because the moon reflects UV, and so predators that have really, that are out at night, that have sensitive eyes and can detect UV, will see these glowing millipedes and know not to mess with them. So once again, a lot of these have both diurnal and nocturnal defenses or aposematism. All right, so that's enough about defense. I'm going to get into something that's really near and dear to my heart as a taxonomist, which is the concept of microendemism. An endemic species is only found in a restricted or defined area, really, really small, usually a small area, but it's only found from one spot, right? So, a lot of animals are only known from Virginia or something like that. That would be endemic to Virginia. In millipedes, however, you get what is called microendemism, where you have species that are only known from one, like a tiny little spot, like one population or a couple populations. A good example is Aphiloria whiteheadi, which you see here. This is the Laurel Creek millipede. It's endemic to an area on and near the Blue Ridge Parkway, not too far from here. And that's the only place in the world you can find it. And millipedes it's really, really common for microendemism to develop in millipedes. And the reasons for that are multiple, but we think, a big part of it is these are flightless, mostly blind. All these millipedes and Zystodesmi are blind. They don't really move around much, so they get really concentrated in one one habitat. And then they also, as I said, don't have that waxy epicuticle, so they don't move around too much. They're kind of restricted. Once they get into a nice gully that's moist in the Appalachians or some swamp somewhere, that's the only place they're going to be. They don't really spread out very often. There are some exceptions. There are some species that are widespread, but the vast majority of these millipedes are pretty much microendemics. And so they have micro-range endemic I have here. It's a species with a distribution of less than 1,000 kilometers squared. 1,000 kilometers squared sounds like a lot. That's about 2 or 3 counties. It's not a huge area. And so for an organism, it's a pretty restricted range. And the majority of millipedes likely are of species are micro-range endemics. For example, 91% of the species in the genus Brachycybe, which is one of the more speciose genera, are MREs. They're only known from a few spots. So conservation efforts are really important for micro-range endemics because they have such small ranges and are so tightly bound to their habitats. They're extremely susceptible to any kind of habitat alteration or climate change or anything that would mess up their habitat. And one thing that's really important to me is this concept of taxonomic anonymity or anonymous extinction, which is that because these millipedes they're like a species can only be found maybe in a certain gully in Appalachia, it may go extinct from pollution or climate change or the digging of something, building of a house or something like that, without us ever learning about it, without science ever discovering that millipede. And the problem with that is that taxonomy is in a lot of ways like trying to build a puzzle. We're trying to understand how all these species relate to each other and how they've evolved over time. And when you have a big, gigantic puzzle that you don't know the number of pieces for, and you don't have a reference picture, it makes it really hard to complete that puzzle when you're missing pieces and you don't know it. You don't know that there are pieces missing. So it's really, really bad. And of course, ethically, it's just bad for the species to be wiped out. But for science, it's also really unfortunate when we never learn about them. And so they'll be gone forever and we'll never have discovered them. So that's something that really drives what I do as a myriapodologist. All right, so I'm now going to quickly run through the 10 orders that you can find in the Piedmont and Appalachia. First are the Polyzenida, which are the pincushion millipedes. The ones are mentioned that have these urticating hairs all over them. So they're like barbed, irritating hairs. They're pretty small, about 4 millimeters, but you can find them. They're really common. You can find them in pine cones and under rocks and things. They have no chemical defenses. They just rely on these barbs, and they eat algae, which is cool. Glomerida are also really tiny. These are the pill millipedes. They look basically like a seed when they roll up. They're really, really tiny. And they're just found in the Blue Ridge. They have this weird structure called the Tomoseveri organ that's a horseshoe shape, and we're not really sure what it's for, but we think it's a humidity detector, which is kind of cool. Then there's the Platydesmeta, which are the feather millipedes. And so this is, one you've seen before, the Brachycybe, which are social. And then there's Andrognathus, which are a little more kind of rustic looking, if you will. But they're found in logs. They feed on fungi. And both of these groups actually exhibit parental care. They take care of their young, which is cool. The Polyzonida are the slug millipedes, which is because they're flat, shiny, and kind of like smooth to the touch, and they're often cream colored. They can be like a nice rose color too. They can get up to about 1 inch long, but they smell like Vicks Vapo Rub. If you ever find these, it's like a really powerful camphor smell. It's kind of nice. But their head is hidden. This is actually their head down here. They've got their 2 antennae, and it's just this tiny little triangular head that's shaped into almost a beak. And they're found in moist leaf litter under logs, like a lot of these. Julida are the snake millipedes, and these are the ones you'll find a lot gardening that kind of thrash around and look like a snake in a lot of ways. They're cylindrical and long. And there's a lot of them are invasive. So Julidae and Blaniulidae, these two families are not native here. The only native family we have is Parajulidae. So you'll find there's a lot of invasive ones. And the males have these, so they hear the gonopods, but males also have these large modified front legs. So that's one way to tell it's a julid. Then the spirobolids are the really big cylindrical ones, which the one we have around here is Narcissus Americanus, which you see here in this photo. That's the ironworm. They can get up to 6 inches long. Well, that's not 6 inches, but like this big. They can secrete chemical defenses that will stain your skin. If you ever pick one up, it can make your skin kind of like a brown, blackish, and it can be kind of irritating. And they also can shoot it. They don't usually, but I got sprayed right under the eye once by one from like a couple feet away, which was a little disarming. But they live in logs. The females are the ones that, I didn't mention the very beginning, I had a photo of an egg in like a cup of mud. Well, that's actually feces. And that's what these spirobolids do. They lay their eggs and then roll them up in poop. And when the young emerge, they consume it as their first meal. And that gives them gut microbiota that can help them break down plant material. Spirostreptida are the regal millipedes. They have these really beautiful structures all over their bodies, and they have a suture running down the body. No, they don't have a suture. That's the Calipodida. That's up next. But these guys have these nice structures all over. They're often purple, kind of purplish color, which is cool. And they have this slight neck. You can't really see here, but their head looks larger than the back of their neck there. Calipodida are the crested millipedes, and they have this suture you can see running all the way down their body. So they look a lot like Spirostreptids. Easy to confuse them. But this is an Abassion, which is brown, fast and common, and smells horrible. Do not, if you see one of these, just don't even bother, because it is really, really foul smelling. But they live in under rocks and logs, and they are kind of adapted to drier areas. And as I said, they have this groove running down the midline of their trunk. Then there's the Chordomatida, which are, they don't really have much, they don't have a common name, but they have these long setae, which are hairs that are kind of hard to see in this picture. They're pretty small, but they have no chemical glands, however, are really diverse. And a lot of them have evolved to cave habitats. So there's a lot of troglodytic cave dwelling chordomatids. And they're found in really moist areas, and they're really fast. They usually run really fast. And then finally, there's the Platydesmeta, which are these, which includes the Zystodesmids, but they are the most diverse order. We have over 500 species just in the eastern US. They have paranota, which I mentioned earlier, which are the extended part of the body segment that allows them to have color on that side and have more of an image to advertise their toxicity. They can be pretty big. They can get pretty large, and they all produce hydrogen cyanide, the entire order. And some are quite colorful, like in the family Zystodesmidae. All right, so I do want to talk briefly about some history of myriapodology in this area, just because it's something that, I assume a lot of you probably have not read much about millipedes or really worried about millipedes, and that's partly due to a lack of research in the area. There are not very many people that have worked on millipedes in the past. The three most important people for this area are Richard Hoffman, who was here at the museum before myself, and he passed away in 2012. Roland Shelley, who is at the North Carolina State Museum, and he passed away in 2018, I think. And then Bill Scheer, who's still working today, very active, still publishing all the time, but he's a really nice guy from Hampton, Sydney. But there's kind of a paucity of researchers in the area, mainly because millipedes are not economically they are economically important in that they break down leaf litter, and if we didn't have them, we'd be drowning in forest debris, but they're not like easily measured. They're not pests, they don't vector viruses, they don't do anything wrong, really. So they're largely ignored for many different reasons. And so that's what makes it so exciting that the Merrick lab at Virginia Tech was created by Paul Merrick, and it's exciting that it now exists. It's been, I think he started in 2013, and it's the only lab in the nation 100% devoted to millipede research. And I was his first PhD student. And so now I am working as the myriapodologist at the Virginia Museum of Natural History in the collection that Richard Hoffman had. And it is, as Tiffany mentioned at the top of this, that it is one of the most important collections of millipedes in the world. And I have some stats to throw at you for that, but we have over 22,000 specimens from all over the world, more than 400 type specimens. A type specimen is, I should explain, is it's very, very important for research. It's basically a voucher. When you describe a species, a new species that you've discovered, and you describe it and name it, you will designate vouchers, which are called types. And those are the specimens that anyone working on that group or that species can look at. They can go to the museum, get that voucher, and compare whatever they've collected with that and be like, oh, it is this species. And so they are basically the representation of that species. And so we have over 400 different types in our collection, which is really, really valuable for research all around the world. We also have over 600 undescribed species and over 200 undescribed genera, which is a lot. You could spend multiple careers just describing the things we have here in the collection from all over the world. But it's a very exciting place to work for a myriapodologist, and I'm really happy to be here. My research focuses, as I've mentioned, on taxonomy and systematics, so a lot of species discovery and description. Some ecology, definitely I do evolutionary work, but I've always been focused on Appalachian and Piedmont millipedes, typically the Zystodesmidae, and now I'm kind of getting into these troglodytic millipedes in the order Chordomatida that I mentioned that are in caves. But I'm also helping set up a molecular lab here at the museum, and we're hoping to work on all sorts of invertebrates like ants, ant systematics, and isopods, maybe crayfish, we'll see. But we've got a lot of exciting ideas, and we're gonna be doing a lot of cool projects in the area. And with that, oh yeah, I wanna mention, as Tiffany said, I am always happy to come out and collect on private property. Private property is really important for us to be able to look at, because usually, one, no one's ever looked at it. And so it's a good place to find new species. And two, a lot of private property is fairly undisturbed, which is really, really nice. You know, you can see a snapshot of the forest, you know, that hasn't been logged. Usually it's been logged maybe 100 years ago or 80 years ago. But it gives you a good idea of like a nice, you know, habitat, and you have a good chance of finding new species. And so if you have any forest with rhododendron coves, especially, or creeks, or springs, or any kind of moist broadleaved forest, let me know. Send me an email. This is my email, jackson.means@vmnh.virginia.gov, and I'd be happy to come take a look. This I want to point out is Sigmoria nantahala from North Carolina. This is the candy cane millipede, and it is one of the most beautiful, coolest looking millipedes in the world, in my opinion. So I just had to show that off. And that, if you go to the nantahala forest, you can find it there. And with that, I'll take any questions that you may have.
Dr. Means, would you mind stop sharing so you can see our group?
Yeah. Sure.
Oh, actually, we won't see our group, but I thought everybody's picture would come on. Yeah, any questions, you can raise your hand and I can allow you to speak, or you can type something in. Here we have something. What is the easy way to tell the males from the females for an amateur?
That's a good question. You know, it's going to be easier on larger millipedes. So the Zystodesmids, the simplest way is basically you flip them upside down, and you kind of hold gently their head in between your thumb and forefinger, peel them back, and look to see if those gonopods are there. So that's the seventh pair of legs. So if you look down the trunk, you'll see leg, and then there's just like these crab claws that are usually very overt. There are some groups like Narcissus Americanus, the ironworm, that have internal gonopods that they have to evert. So you're not going to really see those. However, on those guys, if you see an ironworm on a tree or something, and they have what looks like bumps on their back near their head, that's musculature for the gonopods. That's a male. If they're just smooth, it's a female. And likewise, if you're looking at a zystodesmon, it's just like normal legs all the way down, that's a female specimen.
Any other questions? You can either raise your hand or type it in the chat box. I actually have a question. So in my bathroom, I see this creepy crawly thing with tons of legs go scurrying across the floor. What is the best thing I should do with that? Obviously not stand on it. Where would I take it to?
Well, I mean, it just depends. So if you want to get it out of your house, I would just move it outside. There's not really like, you're not going to be, there's so many of them, not to freak you out, but if you see one, there's probably, 16 others you're not seeing. So moving it outside is the best thing you can do. But unfortunately, you're not gonna get rid of them usually, but yeah, I would just put it outside.
Okay, good to know. We do have a question. Do the invasive species actually hurt the natives?
That's a great question. So that's something we're not 100% sure on, but I will say just in the, nearly, I guess, decade now that I've been working on these guys, or around there, I have noticed an increase in one species in particular, Oxidus gracilis, which is the greenhouse millipede. And it looks like a series of brown beads strung together. That's in the family Paradoxosomatidae, which we don't have here in the US natively. They smell terrible. And they are really, really invasive. They're from China, and now you find them everywhere. Anywhere you go, doesn't matter how remote you are, you'll find them crawling around. And those things we think are competing for resources, competing for leaves. Places you find a lot of Oxidus, it typically has thinner leaf litter, and you're not going to find a lot of the millipede diversity that you're looking for. And so over the time that I've been working on millipedes, I have seen an increase, I think, in Oxidus, and kind of a decrease in the quality of those habitats. But it's something that I know a student was working on years ago at Penn State, I think, but I haven't heard anything since about Oxidus. But we do think that the invasives can cause issues. I'll just say as an aside, one of the big problems for millipedes are invasive earthworms. The earthworms that people have brought in for fishing over the years, they really compete. They eat up all the leaf litter. They turn the soil into this like kind of crackly, dry clay, like mud, and it's not great for millipedes.
Seems we have another question. Within the species that live in microhabitats, can they be crossed between microhabitats?
Do you mean so they can hybridize? If you're asking whether they can, the species can hybridize.
Sonia, if you want to turn off your mic, you can ask that question.
Yeah, so you said that they're really, some of them are very specific to just these small areas, but if if you carried one from each habitat and let them hang out together, would they mate and make new ones?
It's a good question. It's a really good question. Something we've been wondering, especially. So one genus that I'm about to publish a revision on, Nanaria, there's a lot of species. We're describing 36 new species, and a lot of them are from gullies all around Appalachia, and they have these really restricted distributions. And the genetics that the genes that we sequence paint a very interesting, weird picture where it looks like they don't fall out. So they have like really unique gonopods, right? So their gonopods all look different, but the genes do not line up with their gonopod variation. So things that look really similar are distantly related, and things that look really different are close. And that might be due to hybridization. That can be mucking up kind of what we're looking at. But we don't know. That's something we would have to do. We'd have to put we'd have to like target that question, but it is something you can look into, and I'd love to look into in the future with hybridization. I would imagine, a lot of these species have been, just based on our phylogenetics, a lot of these species have been isolated in these areas for a really long time. So I'm not confident that they would be able to successfully mate, especially because of what's called lock and key, where the male's gonopods are in such a different configuration, then that the females might not be able to physically mate with each other. And the females do display some level of choice, where I've seen them, come up to mate and the female will be like, no, and break away and run away. And so the females may not, choose to mate. But that's a good question. We're not 100% sure. And I imagine there's some hybridization. There's got to be.
All right. So the source, who's making the poop where they're laying their egg? What's their favorite poop?
It's mom's poop. So she lays the eggs and then she picks them up one by one. And so she lays the eggs actually, from the Cytopods, and then she brings them down like a conveyor belt down her legs, and then inserts them into her anus, coating them in feces, and then poops them back out. And that is the process. That is the beautiful gift. What can we do?
Okay. One last question. What can we do to help optimize their habitat?
I mean, the best thing you can do is to leave it alone for the most part. They've been here for a really long time, and they do like moist areas. So if you have, just don't muck around with creeks and things like that. Like damming areas might not get rid of them, but it can change the habitat. But I mean, as a homeowner, I would just try to reduce any kind of pesticide use in that area, or any kind of chemical, fertilizers or anything that could, because these millipedes don't have a waxy epicuticle. They're really easy. A lot of chemicals can get in pretty easily through their exoskeleton and their spiracles, which are their breathing tubes. And so I would just kind of leave the area alone, honestly, is the best you can do. But if you want to increase your cystodesmids, planting broadleaf trees is always good. They like broadleaf forests, so maple, oak, things like that.
I think limited use of pesticides is a good practice regardless, right?
Yeah, especially in waterways, if you're doing to a creek. I will also say that hemlocks, a lot of these millipedes are found in hemlock groves, and we think it's probably more of just that hemlocks and millipedes both like the same habitat, not so much the millipedes are attracted to hemlocks, but the loss of the hemlocks to the hemlock woolly adelgid, which is an insect that's killing hemlocks, is really bad, and that increases sunlight and decreases shade in the area, and that's going to reduce moisture in the area and be pretty bad for the millipedes.
And we have a comment that Kathy, you don't rake up all the leaves.
That's a good idea. Yes, leave the leaves. They'll take care of it. Definitely. If you do rake up the leaves, pick up a millipede and bring it to me.
Okay. Good to know. All right. Well, any other questions? Like I said, you can raise your hand, type something in the chat box. Thank you, Dr. Means. Very interesting. I truly learned a lot myself today. I'll be a lot more knowledgeable when I'm hiking through the woods next time.
Good. Awesome. And you can always send photos. You can send photos also to that email if you want. If you see anything cool. Excellent.
Excellent. Well, actually, someone's wanting to see the larvae eating the millipede.
Well, yes, email me and I can send you that video. It's not a great video. I'll try to find a better one, but I do see them unfortunately fairly often. I can describe it as basically it's a decapitated Aphiloria that, head off is still moving, and that's because the fin-goated larva is inside of it. And so you'll see, and at first it looks like, oh, it's just a dead millipede, and then it's like twitching and moving. It's just real creepy. But yeah, unfortunately, I don't have any videos of it like actually, injecting venom, and that would be great to get a video like that. That would be a good find. But
well, and we can't forget that this is lunchtime, so some people
will
still be eating their lunch. Again, thank you so much for joining us and giving us your time this afternoon. Definitely very interesting. And thank you everyone for joining us today and being here. DARPA will be doing several more Lunch with DARPA webinars throughout the year. Definitely, hopefully, you are all getting our e-newsletter. If you're not, you can get that from our website. And thank you. Thank you again, and enjoy this beautiful day, at least we're having here in Virginia and North Carolina.
Thank you for having me.
Thank you, everyone.
Bye.